So we're going to be moving on. The next presentation was listed with Emily Bick, who is a colleague of mine. This is a project that we've worked on together, and she currently is statewide. So Emily Bick is a PhD postdoc fellow, Department of Plant and Environmental Sciences at University of Copenhagen. She is currently stateside interviewing for a professorship. So I'm jumping in and I'm going to give this presentation today. Again, this is work that we've done together. You guys already know them, Cindy Kron. I'm the IPM advisor for the North Coast. So how to use a model to time tilling intervention to disrupt three-cornered alfalfa hopper life cycles. So we all know where it all started with being able to show, Brian Bader was able to show that three-cornered alfalfa hopper was able to acquire and transmit the virus in a greenhouse study. And from there in the beginning, there was a lot of questions. A lot of growers had a lot of questions, understandably so. We didn't know a whole lot. We did know that three-cornered alfalfa hopper's scientific name is Pesticide Applications. I'm going to refer to him as Tcah throughout this presentation. Is a vector of grapevine red blotch virus in a greenhouse study, native to the US, so it is not an invasive species. It is an insect that's been here that is native to this area. It was studied predominantly on alfalfa, peanuts, and soybeans in the southern United States because they're major pests of those crops. And I should point out while I'm at it is that those crops are all legumes, so something to think about later on in our presentation. It was previously considered only an incidental pest of grapevines because it would girdle petioles, and very little was known about its association with vineyards. So in the beginning, again, there was like so many questions, and so here's some of the questions, but today I'm going to focus on two of them that are going to guide us as to being able to predict when to till under cover crops. So we try to approach the questions of what are their hosts and their preferred hosts, and when is Tcah most vulnerable to control methods. So this is a few papers that came out of my dissertation research, and I'm going to start with focusing on these two papers that were looking at feeding and reproductive hosts of three-cornered alfalfa hopper. And out of that work, this is a table that's in the American Journal of Enology and Viticulture, in which we compiled my research and previous research that looked at feeding and reproductive hosts of three-cornered alfalfa hopper. And in this red box, I want to point out this is all Fabaceae. Fabaceae are legumes. So it is the family of legumes, and every single one was feeding and reproductive hosts. And when I say reproductive hosts, it's able to lay eggs and nymphs emerge. We did not look at how well that host is able to allow the insect to go from an egg to an adult, just if the eggs are laid and if nymphs emerge. But some of these hosts are ones that we use and currently use to rear three-cornered alfalfa hopper on in the greenhouse for studies. So from there, we're going to go back and focus on this next paper looking at seasonal dynamics. So we know that three-cornered alfalfa hopper preferred legumes from previous research and from the research that I conducted. Every single legume that we tested, which were 13 of them, were hosts. So now we're trying to address the questions of seasonal dynamics in the vineyard. When do they arrive in the vineyard? How many generations do they have? Do they overwinter in the vineyard? There was a lot of those kind of questions that we were trying to focus on. And so this study was done at Oakville Research Station. This is the vineyard block and the irrigation pond. I sampled around the irrigation pond, and every other row was tilled under. So I sampled every other row that had ground cover, 26 rows, and the perimeter. It was a little bit more than a mile distance each week, every week for 2 years straight, including winter. So what we came out of our samples, this was done by SweepNet. And this is the data that came out from 2 years worth of sampling. So in green, you can see are the nymphs that we found, and blue are the adult three-cornered alfalfa hopper. And what we show is that three-cornered alfalfa hopper arrives in late winter, and this is the immigrating overwintering population, comes into the vineyard, it lays its eggs, and it proceeds to have a depression in the population. And you see this depression here, and you see this depression here. And what's happening is that the three-cornered alfalfa hopper has overwintered, and it takes a lot of energy to be able to overwinter. And when it comes into the field, it has enough stored fat body and energy to create the eggs, lay the eggs, and then it dies off. And that's a pretty normal behavior in insects. It's completed its life cycle. It's done what it needs to do. So the nice thing is that during this time, there's a depression in the adult population, and this is the period of time in which the nymphs are developing on the ground cover. And then all of a sudden, we see this jump here, and we see the same jump here after the depression the next year. And what's happening is that those nymphs that are developing during this time are now emerging as adults. The problem with the sampling was, first of all, we found very few nymphs throughout the whole trial. And the first day that we find nymphs, both in 2016 and in 2017, was the first day that the adult population jumps. Now, ideally, you really want to go after the immature stages. And how can we predict when those immature stages are if by the time we see them, the adult population has already emerged? Now, we have this mapped out for Oakville, but temperature drives insect development. Warmer temperatures to a certain degree would mean the insects would arrive as an adult sooner than temperatures that are cooler. And because the wine grape growing region has a range of temperatures throughout the season, you cannot necessarily apply this graph in Oakville to, say, Carneros. The insect development, you wouldn't expect to be exactly spot on the time. And there's also seasonal changes in temperature. Some years are cooler than others. Some years are hotter than others throughout the whole growing region. So the problem we were having was sweep netting. As you can see, these are pictures of the immature stages, and the first instar on the left and the third instar in the middle are very small. They range between 1.4 to 3 millimeters. They're clear. They start out clear like this, and then they turn a sort of a straw color. Well, those are very hard to see in a sweep net sample. And remember, we're processing 84 samples a week in the field looking through a sweep net. So it's very difficult for you to see these, and for a grower too. If a grower were to go out there and sweep, are they likely to see them in their sweep net to count them? So it wasn't effective. We were able to see the fifth instars. These photos here are of fifth instars. They come in 2 different color morphs. They're larger. They're 3.6 to 5 millimeters. They're chunkier. They have more coloration. They're very noticeable in a sweep net sample. So, and here is a photo to show you, like, this is a typical sweep net sample. This vineyard actually had a lot of aphids, but there's all kinds of different insects. There is plant material. So you're trying to find these really tiny, clear to straw colored insects within this kind of sample. And so this was the dilemma that we came across. Okay, so like, what do? We want to pinpoint this area in which the three-cornered alfalfa hopper is present on the ground cover and immature. And I guess I should add the reason why. Why is it then? Well, immature stages don't have wings. They cannot fly away. They don't disperse long distances. The fourth and fifth instars will move maybe to the plant next to them, but they're not moving 10 feet. And the first through third instar tend to stay at the base of whatever plant that is their host. So they are very vulnerable to removing their food source. So if you were to till underground cover at a time in which they're developing on that ground cover, and that ground cover were to die, the plant material, the insects don't have the ability to disperse to find other food sources. And so that is why we're going after the immature stages. So here we are, this dilemma of, okay, we know that they're going to be here before the first generation emerges, but how do we predict this to figure out when is the ideal time to till under your ground cover? So in addition, you'll see the graph on the bottom. I don't know why it keeps moving. The graph on the bottom, it shows you 1 year of our adult data. So this is in green is our number of adults. And in that year, in a subset of the same block, we sampled girdles. There was the question of, well, okay, well, when are three-cornered alfalfa hopper girdling? When could they be transmitting? And so as you can see in the graph here, girdling started about mid-June and increased over this season. And as you see the adult population dwindling on the ground cover, which we were sampling, girdling was increasing in the canopy. So what is going on is that it's summertime, ground cover is not watered here. We don't do flood irrigation. So the ground cover is dying off, which is their host. And the adults, when they emerge, they go up into the canopy because it's their food, it's a source of food that they have. They no longer have, for the most part, the legumes available on the ground cover for them to feed on, which is what they would prefer. But, you know, grapes are right there. They have nitrogen in them. Insects like nitrogen. And so they'll move up into the canopy and start to feed. And so that is what you're seeing here. And I included a graph from Sinowitz et al., which is Mark Fuchs' group, that shows, they also showed in a vineyard a couple miles from my research vineyard that three-cornered alfalfa hoppers started to test positive in June, which is when I was able to show that three-cornered alfalfa hoppers started girdling in the vineyards. And so it is at this time that we believe in that movement that they're allowed to, you know, feed on an infected vine, they can acquire the virus and eventually transmit. So really, it is this time of immature even more so that we want to attack and go after. And so the next paper is I worked with Emily Bick on to create a model to determine when is the ideal time to till under cover crop. I am not a modeler, Emily is. So she took my data and created a model to help try to predict when is that ideal time. So what you're looking at here is what the model is showing. So I don't know why it keeps doing this. I'm gonna leave it alone. So in green is the simulated small nymphs. And so we broke it up between small nymphs and large nymphs. Large nymphs, if they're like in like the 5th instar, they might, if tilled under, the ground cover tilled under, they might have enough reserves to actually emerge as an adult without continued feeding. So, and it does take, when you till under ground cover, it does take some time for that ground cover to actually truly die off. So the small nymphs we have as 1st through 3rd instars, they're the most susceptible. And in this graph, you can see in green when the model predicted the small nymphs will be present, and then it shifts to larger nymphs, and then it shifts to adults. So we expect to initially find adults from the first infield generation between 1743 and 1915 accumulated degree days, and that's in Celsius, not in Fahrenheit. So that's important to note for later. And that area is here highlighted in yellow. And so that's the time frame in which we expect to find our first field generation emerge. And so tilling recommendation is here, this area in red, according to the model. And that indicates when 25% to 80% of the small nymphs are present in the field. And this is really where we want to focus on going after. So the this supports the recommendation for tilling between 1310 and 1565 accumulated degree days, highlighted in red. So this information came out of the model of when is 25 to 80% of the immature stages in the field, and when would I have the most bang for my buck? Some people would say like, oh, well, why don't you just till earlier? And the thing is that if you have legumes in your ground cover, or you plant, say, a cover crop with legumes, the longer those legumes are in the field, the more nitrogen they're able to accumulate and add back into the soil when tilled under. So tilling too early, you're losing some of that benefit, but still paying the cost of putting those legumes out there in the field. So what we did, so we have this model that we created from data from here, Oakville Research Station, and we took the second year of data to create the model, and then the first year data to validate that model to show that, yes, it does work. We're able to predict this window. In 2020, you can see the marked in green were 3 sites that I sampled, and I sampled starting in the beginning of February to see them come in the field. Then there was the depression, and then sampled until the first adult emergence. So we were trying to pinpoint the first adult emergence and to see if we could use the model in that area, and it would accurately predict when that was going to occur. In 2021, we sampled 5 other regions because I'm trying to show, okay, can this model work not just in Oakville, but outside of Oakville in a range of temperatures. Unfortunately, last year was extremely dry, more dry than I've ever seen, really. Drought was really bad this year. And so normally when you go out to a field and they mow, the ground cover bounces back until a little bit later in the summer where it's too dry and then starts to die off. This year it was so dry that when they mowed, a lot of vineyards, the ground cover did not bounce back and everything was dead. And so this happened in 3 of my locations. We were not able to show the first generation emergence. There was nothing to sample. It was all dead. I tried to sample it, but there was no adults. We couldn't indicate when that emergence happened. So what we have is the original location and 5 other locations in which we were able to show that the model worked, that we're able to predict that period of time. So I've given you this information. What does it all mean? How can you use it? Here is UCIPM's website, and you can see here to the right, ipm.ucanr.edu/weather/index.html. You go to this web page, and this is how you're going to use the model if you want to predict when to till under. You go to this website, you go to the page, you scroll down this page to view the degree day calendar. So if you scroll down to the bottom, this is what it looks like. And you're going to click to choose Celsius, and you start to enter the lower as 0. So we're starting at 0 to Celsius, and then you press calculate. The next page takes you, choose your location. So I'm in Sonoma currently. I chose Sonoma. You choose the start date January 1st of your current year that you're looking at, and your end date would be your current date. So when I did this, it was April 27th. I chose that as my current date. And then you click continue right here. And that's going to take you to a page that gives you options of weather stations. I chose Santa Rosa. My office is in Santa Rosa. I click Santa Rosa. And that takes me to this page where you put air temperature. This is automatically formatted where it says air temperature and formatted report already. So you don't need to do anything but click calculate button. And what that does is spit out your accumulated degree days in Celsius. Very important that you choose Celsius in the beginning, because otherwise you're not going to get the right numbers. And you go scroll down to the days in which the degree day value is between 1310 and 1565. So you want your current, when you're looking, you know, say it's in March, and you're like, okay, where am I degree day wise? Well, you can see as you're approaching, 1310 would be the lower end and 1565 is the higher end. And so you want to try to find a time that you can till under during those degree days, 1310 to 1565. And so that is what this website will do. It'll help direct you to that right range that works for your specific area. And I will be posting video slides of how to do this step by step on my webpage, which is ucanr.edu /NCIPM. So I have all kinds of newsletters on that page, but there will be links shortly to be able to guide you step by step of what I just went through, in case you want to pull it up anytime to do for your specific vineyard. If you have any questions, my email address is ckron@ucanr.edu, Twitter handle, and then the website where you're able to enter this information to get your degree day calculation. Again, is ipm.ucanr.edu /weather/index.html. And with that, I will take any questions.
Yeah, so there is one question. Is mating disruption an option for TCAH?
Not to my knowledge. I don't believe they use pheromones to attract each other. I do know that they do some movement with their feet on the substrate to attract a mate on the same plant. That's been looked at, but in order to do mating disruption, you would have to pinpoint a pheromone that is an attractant, and I don't believe that they believe that this insect actually uses that.
Great, and then there was a question from 1045. So at the start of the presentation and kind of related to the previous presentation, for you, the follow-up question is that theoretically, if you've maintained ground cover of non-host cover crops, you wouldn't benefit from disking. Is that correct?
So three-cornered alfalfa hopper has a range of hosts. Previous research has shown that they're attracted to legumes. And if you're sure that there are no, if it's all non-host, you likely aren't supporting a population. The insect needs to feed on a host. So there are plants that it won't feed on. And if that is what you have in your field, the insects will continue to move on and not be attracted into your vineyard. So yes, but that's a rare occurrence that you have a stand of just non-host, because there are a lot of wild or escape vetches and clovers out there that can support, but also too, they're leguminous weeds like burr clover and Spanish clover that were never planted, that are throughout a lot of vineyards. But in theory, if you literally have no host in your vineyards, you wouldn't be finding three-cornered alfalfa hopper there. They would be moving on to find something to feed on.
Another question, it sounds like dead TCAH cannot survive even on dead cover crops. It's a little heavy-handed, but could chemical mowing be a viable option to mitigating TCAH populations?
When you say chemical mowing, are you saying applying herbicide to the whole ground cover? Or are you saying applying a pesticide to the ground cover that would go after the immature stages?
They said yes, herbicide, I believe.
Oh, so I could never recommend broadcasting an herbicide across all the ground. Environmentally, it would not be it would not be good for the environment. And as an IPM advisor, I could not say, yes, go apply a broad herbicide to the whole ground cover of your vineyard. Would it work in killing the plants? Yes, but there's a lot of things that would work that are not good for the environment. This is why we were trying to look at, okay, can we pinpoint a weak part of the development of this insect to be able in a natural way, or more of a natural way, combat the populations, or at least make your vineyard not as attractive. As far as when it comes, someone earlier was asking about pesticide applications, and there are a few issues with pesticide applications that I wanted to address, because I do have a few minutes. First of all, research in the southern United States on peanut, soybeans, and alfalfa, where they are a major pest of these crops, do not have a good response to pesticides. They have not been able to find pesticides that are very effective in controlling the populations. So there's that. Second, you know, if you are going after the immature stages that are mobile, you can't spray a pesticide on the ground cover. It's not registered on random ground cover, so it's not legal. And then if you're going to spray in the canopy, the issue is that the insects are mobile, and they are not only just on the ground cover and the canopy at the same time, the adults both. So you're not going to get effective control. And because of what we know about this insect, there aren't products that really are known to effectively control it, even in major crops in the South. So pesticide has never really been a viable option when we looked at this insect. It's not very susceptible.
There is another question about what degree days are.
Okay, it's accumulation. So we started the model where it's at 0. And the model was created so that we could start the accumulation at 0 degrees Celsius. Typically, you would start accumulation of degree days at the lowest threshold of insect development for that insect. So if that meant the insect would stop developing at 54 degrees, but at 55 it would develop, then anytime there is a temperature above 55, you would accumulate these degree days. And it's a little bit more difficult to explain in 1 minute. So I can send you the information. Is this Brooke? I can send you the information of explaining visually what I'm talking about. It's an accumulation of the amount of degrees in which the insect can develop, and how that adds up over time. That is able to pinpoint this period of time in which the development should be at a certain stage. And I will send you more information, Brooke, on how that is calculated.